is Cydonia oblonga, NADES, ultrasonic assistant extraction and food waste valorization. Green valorization of Quincy waste using natural depth authentic solvent and ultrasonic assistant extraction, which is the main problem. The main problem is the waste generation. A large amount of waste generation by Quincy and JAME industries, rich in polyphenols and antioxidants. This fraction traditional extraction, because I'm studying with compare traditional extraction and green technology of or green extraction. Traditional extraction, toxic organic solvent used, high energy consumption, and low stability. Other side, green extraction in key studies, NADES and ultrasonic assistant extraction, natural and non-toxic solvent use, enhanced recovery of bioactives, reduced energy consumption, and environmentally friendly. I'm of the study to evaluation of efficacy of natural deep authentic solvent combined with ultrasonic assistant extraction for recovering polyphenols from Quincy waste. Specific objectives: develop of a new green solvent of NADES system, choline chloride and glycerol. Compare NADES ultrasonic assistant extraction, green technology, with conventional 65% ethanol extraction or traditional extraction, solvent extraction. Assess potential for total phenolic content and antioxidant activity by DPPSD and ABTS-HE. Assess potential for nutraceuticals, functional food, and circular bioeconomy application. Experimental procedure, sample preparation. The first, dry the Quincy peel sample, chop into the small piece. After that, added NADES, mix choline chloride and glycerol to form a natural deep authentic solvent, sonification in 55 degrees Celsius temperature for 30 minutes. And after that, measure total phenolic content and antioxidant activity by visible spectrophotometer. Total phenolic contents and antioxidant activity compare traditional solvent 65% of ethanol and deep natural authentic solvent, and the amount of total phenolic content in a deep natural authentic solvent in 2 more high of extraction with traditional solvent. And antioxidant activity, strong correlation between total phenolic contents and antioxidant activity. Conclusion and relevance to food waste stop, key finding. NADES ultrasonic assistant extraction is the effective green extraction technology, significantly increase the recovery of polyphenols and antioxidants, reduce solvent toxicity, energy consumption. Future perspectives, scale-up studies, toxicological evaluation, and industrial implementation. Contribution to food waste stop goals, waste valorization, sustainability food system, circular bioeconomy, and future perspectives, scale-up studies, toxicological evaluation, and industrial implementation. Valorization and potential application of Quincy waste, functional food, natural antioxidants, nutraceuticals, and including circular bioeconomy. Thank you for attention.
Thank you very much for that very clear and concise talk. It's very nice. Do we have some questions, please, in the audience?
Hello. Hello?
It was. Did you perform any optimization of extraction? Because for every raw material you use for extraction, you should have an optimization because the parameters for extraction depends influence
Yeah.
of polyphenolic content and antioxidant activity.
Yeah, in this study or key study is a short communication and short presentation. And continue in future, this study, we optimization parameter, temperature, solvent ratio, and other parameter by surface methodology or simplex lattice methodology or other advanced statistical approach. Thanks.
Thank you. Are there any more questions? Please.
Thank you for your presentation. At what stage of the fruit development do you extract the polyphenols? This is one. Second, what happens if you prepare jam from the fruit? Are the polyphenols still there in a good way for eating them and consuming them for the health of the people?
Yeah, but in key studies, the aim studies is to valorization waste of quince and other. But during a processing of fruits and vegetable, a generation more waste. And I must study from waste generation to extract polyphenols and after that measure antioxidant activity between a strong correlation between phenolic content and antioxidant activity. Our interest is to valorization waste.
Thank you. Are there any more questions? No? Okay, thank you very much again.
Thank you.
Okay. So our next speaker is Mario Riolo, who's a postdoc researcher at the University of Catania in Italy. And his talk is a citrus byproduct bioformulation, BioAct LM, to control blue mold and brown rot in post-harvest value chain. Thank you, Mario.
Okay, good morning. Thank you for the nice presentation and thank you for the nice presentation and thank you for the opportunity to present within a construction food waste stop. The main idea of this study is simple. We take a citrus side stream and we test wherever it can help to reduce post-harvest loss in the same value chain. In this talk, I will start with the rationale and objective, then I will show you how bioactive lemon medium is produced, and finally I'll present the result of 2 case studies and the main implication. Okay, post-harvest rots are a major cause of loss in citrus. Even small wounds during harvesting, grading, and handling can become infection point. Then decay can develop quickly during storage and shelf life. At the same time, control options are limited, so it's very important to find a new solution and green solution. This study, in parallel, citrus processing generates large amount of peel, and this peel remain underused, so they are a clear target for the valorization. This study is aligned with the objective of Food Waste Stop, and our aim was to develop a new formulation called bioactive lemon medium to reduce post-harvest loss and fit a circular economy approach. We assess bioactive lemon medium through direct contact assay, bulk mediated assay, and in vivo fruit test. The question is practical. Can a pill powder formulation contribute to reduce post-harvest loss in the same value chain? And we started from, I will describe briefly the production of lemon medium because I don't have a lot of time, but we started from a lemon peel powder, a typical citrus byproduct, and we use 2 strains of Lactibacillus plantarum in order to fermentate our formulation with each strain. The fermented matrix contain several fermentation metabolites and citrus compounds. And we decided to test firstly in 2 ways. Direct contact assay in order to evaluate the inhibition in vitro, and then with the volatic mediated assay activity. Before to move with the case study, I will show you briefly how we select these 2 strains. We started from 13 lactic acid bacteria isolate recovered from citrus peel, and we identified them by MALDI-TOF. Then an initial in vitro screening was performed using a dual culture overlay and agar diffusion test. And the pathogen panels used in this study includes fungi and homicides such as Penicillium species and Phytophthora species in order to provide a large broad of pathogen tests. At the end of the initial screening, we found 2 isolates of Lactibacillus plantarum that showed a digest and consistent inhibition across the pathogen panel. So the next step was to use this bacteria for the fermentation of lemon peel powder. And the next question was, what is in the fermented matrix, and what are the main differences between the 2 formulations with each strain? And from a chemical point of view, we found mainly organic acid, including lactic and acetic acid produced by the Lactibacillus plantarum strains, and also minor compound such as salicylic acid, phenolactic acid belonging to the phenolic acid. Also, bioactive lemon medium with gas chromatography showed several volatile organic compounds, and overall, this is a multi-compound mixture, and the 2 strains showed a different chemical profile. So we decided to test both formulations in the first case study to control the blue mold on oranges. So in this case, we inoculate Penicillium digitatum, which is one of the most important pathogens in post-harvest in citrus. And we tested both formulation and compared with control. Both treatments showed disease reduction. Sporulation was reduced by about 2 orders of magnitudes compared with the control. So the bioactive lemon medium reduced the blue mold and improved the shelf life of the fruit. In the second case study, as in the first case study, before we quantify the minimum inhibitory concentration of bioactive lemon medium and the minimum fungicidial concentration against several Phytophthora species. And we tested the bioactive lemon medium ranged from 0.6 to 300 milligram ml, and we observed a species-dependent response depends the species tested. And the minimum inhibitory concentration ranged from 1875 to 150, and the minimum fungicide ranged from 37.5 to 300 milligram. So after we decide to evaluate the VOC-mediated inhibition using a sealed space assay, so without direct contact. And also in this case, we found a species-dependent inhibition with inhibition of Mycelia grow ranging from 33% up to 100%. In the case of Phytophthora pseudosyringe, this isolate was fully inhibited. Then we decide to test bioactive lemon in vivo, and let's look at in vivo test. At the top, you can see the representative symptoms of treated and untreated fruit on oranges and apple. And also in vivo, we found a species-dependent efficacy of bioactive lemon medium with inhibition ranging from less than 35% up to more or less 65–70%. At this stage, we found three main limitations. Packaging house protocol is not yet optimized, so we need to define dose, contact time, and mode of action of bioactive lemon medium under realistic condition. Second, the efficacy depends on conditions such as cultivar, wounds type, storage, and infection pressure. Third, we need to control batch-to-batch variability because fermentation and pill batches can change the final composition of the formulation. The next step are practical. We define dose and contact time and mode of action, and we are looking for stability and compatibility with coatings, waxes, and also the use of sensor in order to evaluate abiotic factor that can affect the activity of bioactive lemon medium. And finally, we move to semi-commercial validation and regulatory pathway. At the chain level, we found three main entry points. The first is the packaging house. Bioactive lemon medium could be used as a post-wash application to target early infection. In the cold storage, may support disease suppression and reduce cross-contamination risk. This is especially relevant for the blue mold and the green mold, where the sporulation can drive secondary spread in the storage room. And finally, as a formulation route, because bioactive lemon medium could be used as a standalone liquid treatment or high ingredient for biodegradable coatings. Full experimental details are reported in these two publications. The first one related to the blue mold control using bioactive lemon medium. The second one related to the anti-omycete potential of bioactive lemon medium against several Phytophthora species. Let's take me close with these four take-home messages. First, bioactive lemon medium is obtained by fermenting lemon peel powder with Lactobacillus plantarum strain from the citrus peel. Second, bioactive lemon medium reduce blue mold and brown rot in fruit assays. We observed two complementary effects, contact inhibition and VOC-mediated inhibition. And finally, this is aligned with the food waste stop themes, so loss reduction and citrus byproduct valorization. This is one of the last articles published from our group in collaboration with Professor Barbara Conti and Professor Romanazzi within Cost Action Food Waste Stop. Acknowledgement to my research group coordinated by Professor Cacciola. Thank you.
Thank you very much, Mario, for your presentation and for keeping the time. Do we have any questions from the audience? Yes.
Thank you, Mario, for this interesting presentation. I was wondering, why did you choose to study Penicillium italicum in this case instead of Digitatum, if there is any reason, or maybe you already got results which you have not shown here today?
Okay.
And the second thing, have you seen what's happening with the limonen in the pill after the fermentation process? Because limonen is a volatile in the citrus pill, which is very, very important for the pathogenicity of Penicillium species.
Okay, about the first question, we evaluate the activity against the green mold and the blue mold, but we prefer to focus on the blue mold because the temperature of sporulation of Penicillium italicum are more lower compared with the green mold caused by Penicillium digitatum. So in the first step, we evaluate the activity focused on both pathogens, but after we evaluate the chain level. So in the storage room with lower temperature, then we move this fruit at the temperature of 15 degrees, more or less, and after in 25 degrees. So in this way, we evaluate all chain of citrus processing from the field under the supermarket. About the second question, we started initially with the evaluation of the peel polder without bacteria. So we characterize the profile in order to know the property of and the volatic compound with gas chromatography present on the lemon peel polder. We also evaluate the profile, chemical profile of the oranges peel polder. But at the end, the best was the lemon peel polder because there are a lots of compounds that are perfect for the fermentation with the Lactobacillus plantarum, because it's not easy ferment the bacteria with another byproduct compound. For this reason, we used also a minimum medium, and we added the lemon peel polder. So it's not only lemon peel, water, and bacteria, but there is a minimum medium with specific component in order to stimulate the growth of the bacteria. I don't know if I answer. Okay.
We have time for more questions. Yes. We have one at the end of the room.
Thank you very much for your nice, good work. My question was, okay, you haven't, characterized the fermenting medium of Lactobacillus plantarum. However, have you looked at the liability of the bacteria? Because the bacteria itself may, contain some of the bacteriocins, within the membrane also, and some kind of enzymes, and also some antifungal components, and they may, still express on your liquid phase. I mean, in the spray form. Mm-hmm.
We firstly evaluate an initial in vitro screening with all bacteria. Firstly, with the overlay using only the bacteria, then with the fermented bacteria on MRS. So we evaluate with agar diffusion test the capacity of the metabolites produced by each strain, each isolate of Lactobacillus plantarum, but not only Lactobacillus plantarum was characterized. But however, we evaluate firstly the activity of each. Then we select the two most consistent bacteria that inhibit our pathogens, because not only Penicillium, Phytophthora, but also Fusarium, Mucor, Aspergillus, a lot of icebergs and genera.
Let us take some of the antifungal compounds in the postbiotic.
Exactly.
Thank you.
You're welcome.
Okay, thank you. We are leaving the eventual other questions for the coffee break. So thank you again, Mario, for your presentation. And I invite now on the stage the next speaker, Sar Tanner from Sweden, a postdoc researcher at the University of Borås. And the talk will be about turning pomegranate byproducts into sustainable protein source.
Hello, good morning. I guess we continue with the fruits. And in this presentation, I want to talk about pomegranate waste materials, how we can use in the food industry. Can you imagine for this good page and picture and what the behind of this picture in the industry, we have so much waste materials. For example, is pineapple, orange, or the pomegranates. And we want to choose the pomegranates as it's very common in the Europe, especially in the Mediterranean area. Also Turkey, Iran, and Afghanistan. This region is very commonly used. Not only the growing under this area, also, for example, in Sweden, we don't have any pomegranate, but it's coming from the Turkey or Cyprus and processing in the country. So it means the waste material has been in the country also. So it is problem for all countries. Pomegranate is very common in the world. For example, is the pomegranate juice and also pomegranate sauce, vinegar or the wine. So we have different type of the materials in the world. For the characterization, pomegranate, we eat just only the seeds, but the outlier is like a waste material or byproduct, which is that we don't use. And it's also for almost the 40% of the food is the waste material. If we see the contents, so it's very rich in the sugars, especially glucose and the fructose in the pomegranate peel and also in the juice and the barrel. And also it contains some punicologen and other bioactive compounds. These are also as a carbon source for microorganisms. Of course, it's inhibiting, but also they can consume some of them. In this work, we use the 3 different type of the fungi. These are the traditional food in the southeastern Asia, which are the onkom, miso, tofu, or tempeh, and used by the Aspergillus oryzae, Neurospora intermedia, Oligospores, Rhizobus oligospores, and more also, but we use just 3 of them. In this work, this is the 2-stage. The first one is the shake flask experiment and the screening of these 3 types of the byproducts, which are the peel, juice, and aril, and use 3 different fungi. After that, we scale up in the 4-liter bubble column reactors. Yeah, first in the shake flask. First, we try the effects of this juice, how is it affecting? And we use normal glucose and yeast extract media and adding the different concentration of the pomegranate juice. In the first law, Aspergillus oryzae, so it didn't affect so much, so it's not inhibiting. But in the oligospores, you can see it's inhibiting the fungal growth. And Neurospora is not affected, so it gives a similar. After that, we tried just the juice without any supplementation. Firstly, we tried the 4% of the fresh juice. The best is in the Aspergillus oryzae, then following by Neurospora intermedia. The result was a bit affected in this, it gave the lower or lowest biomass. And then we increased the concentration of the juice to 10 to 20%. You can see 15% is good in the Aspergillus oryzae, but after that, the yield is reducing based on the volume of the juice. We compared it with the fresh juice and compare with the commercial. We can think about this. It can be as a expired juice, so how we can use it for the industry. For the commercial juice, it's gradually increase the biomass, but I will say it later. And let's go to the Neurospora intermedia. So if we increase the pomegranate juice volume, so it's also inhibiting, not like Aspergillus. But in fresh juice, it's inhibiting, but in commercial juice, it's not inhibiting, it's gradually increasing, and you can see this yield also is also increasing. In the glucose and yeast extract in the media, the protein contains around 40–50%, and is not affecting or inhibiting the protein accumulation. But in the without the Supplementation is interestingly fresh juice. It gives the high protein is 15%, but in commercial juice it gives the almost 4%. Something is happening in the industry, so we can't use the commercial juice for the protein productions. Then we tried to effects of the aryl again in the glucose and yeast extract media. Actually, I can say it's not successful because in this state there is a white particles. It's not dissolved exactly in the liquid media. So when we when we cultivated the fungi, almost we harvested some this white thick material. So we have some data, but I can say that it's not for fungi; it's for the harvested solid. So aryl is not useful for the fungal protein or fungal production. Then we pass to pomegranate peel, again using the glucose and yeast extract media for screening the effects of the peel. In Aspergillus aureus, you can see it's very nice and gradually increasing depending on the increasing of the peel concentration. Rhizobus oligosporus now is not affected in glucose and yeast extract media. Neurosporia intermediate is a bit weird. So in the low concentration is a bit inhibiting, but after that is increase the volume. Of course, it's harvested solid because we use the pomegranate peel. It's not totally dissolved in the media. But now we can use without the supplementation just for the peel. And first we tried effects of the pH. The Aspergillus oryzea that gives the base condition in the pH 5. And Neurosporia intermediate is 5 or 5.5. And now we can see the Rhizobus did not grow on the pomegranate peel without any supplementation. So it's totally inhibited. And the B graph, you can see that we compared the just harvested solid and dry peel and the wet peel. We thought maybe there are some volatile compounds can be affected on the fungal protein, fungal growth production. So that's why we use the wet peel directly. So in the Aspergillus, it's almost gave the similar, it's the standard deviation. Neurosporus gives the similar. As the Rhizobus oligosporus is a bit grown better in the wet peel. But in the industry, if you think so, the wet material can be used. It can be difficult to use it. When we come to the protein concentration, the harvested solid is not fermented, it's just dissolved in the water, and after that harvested, it gives the 6% protein, but in the fungi production also it's almost to give the same results. So it didn't improve the protein production. To improve the production in protein production, we supplemented with different nitrogen sources. The first is yeast extract, urea, sodium nitrate, ammonium sulfate, and ammonium chloride. The Aspergillus oryzea almost give the same levels regarding the fungal or harvested solids, but in the Neurosporia intermedia, the urea and the ammonium sulfate gave the highest number. But regarding the protein contents, in the both condition, the yeast extract supplementation is the best. Then after that, we choose to this good condition in 5 gram per liter yeast extract and with the 40 gram pomegranate peel, and we tried in the 4 liter bubble column reactors. Then after that, we harvested almost 20 gram per liter harvested solids. Then we characterize it also. So the fungi can accumulate a bit total fats, and also for protein is improved. And also it contains the crude fibers. You can see the glucose and fructose mostly consumed in the fungi. So we can say the Aspergillus and Neurosporia intermedia were resistant to effects of the pomegranate juice. Aryl is not good for the fungal growth, and yeah, the peel also can be usable for the fungal protein production. So when we consider the one ton pomegranates, if you use them, we can produce almost 500 liters pomegranate juice, and 440 kgs goes to the peel as a waste material. So it can turn to 140 kg biomass production. So yeah, my presentation is like this. Thank you.
Thank you very much for the very, very nice presentation. Do we have some questions in the audience, please?
Sorry. It is after the harvesting the material is just for the before the drying is wet fungal.
Is it with the fungal
Fungal with the pomegranate peel. Yeah, it's like a meatball.
Have you tested the effect for the potential mycotoxin production of these strains?
No, not yet. But of course we should check it. Yeah.
Yes,
Yes,
but
for
maybe.
Aspergillus oryz especially.
Thank you.
May not nurse for this.
Any more questions in the room? No. Okay, then, well, if you
Thank
think
you.
of questions, you can ask him later. So moving on to our next presenter, it's Adnan El Yacoubi, who's a professor at the University Sultan Moulay Slimane in Morocco, representing the Association Green Development and Innovation. And his presentation, which is very close to my heart, it's From Waste to Value, the use of black soldier fly larvae in agro-food waste management to produce protein and promote circular bioeconomy.
Good morning, everyone. I'm Adnan El Yacoubi, professor at University Sultan Moulay Slimane, but also president of the Association Green Development and Innovation. In my presentation today, I will focus on the use of the potential black soldier fly in converting organic waste into valuable resources, such as producing new alternative proteins, which is used for animal and human feed and food, and also in production of frass as a biofertilizer for crop growing. So as you know, the main problematic in Mediterranean agriculture is urbanization pressure on resources, climate change increasing year after year, inefficient organic waste management, food insecurity caused by urbanization pressure and climate change. Also, a lack of policy and governance that can reduce efficiently the organic waste management even in urban and rural area, and also the limited integration of digital tools in the process. At global scales, for example, the fish and seafood sector is the main sector that generates more and high level of food waste. And in the second place, there is fruit vegetables sector, which is producing about 27% of waste. In Morocco, 70% of waste are organic waste, representing about 70 to 90 million tons per year. This waste, organic waste, are coming mostly from agricultural practices, such as crop residue and manure production. This is why insect training could be a powerful agroecological tool for organic waste management, production of alternative protein, and for production of frass, which is a biofertilizer for crop growing. Why insect farming? Because insects are feeding on urban agro-industrial and byproduct and organic waste. They use less quantities of water for producing protein and also small spaces up to 18 meters square to produce 1 kilogram of protein. And also this insect farming can contribute in reduction of carbon dioxide. Also, it consists of safe as a food, feed, and biofertilizer, and it contains highly nutritious and high-value byproduct. What is required for insect farming? We need just a growing room with a controlled temperature and humidity. The temperature is about 27 and 32 degrees and humidity of 60 and 70%. And the substrate for insect rearing with a pH with 60 to 8 and minimal and non-urine rearing. We need also some good cages for incubation of insect and larvae and good ventilation. The substrates required in this process are existing everywhere. So we need only fruit and vegetable waste collected, for example, from market, restaurant and household food waste, and agricultural residues such as straw, soybean, crude, and brewery waste. This is an example of pilot production units for insect rearing. As I said, you need only a growing room in which the temperature and the humidity is controlled, and some dark cage to put inside the larva in pupa stage, and the love cage, this in the left, which is connected with the dark cage, and it's allowed to emerge insect or larva from dark cage to love cage that allow female and male for mating inside. Also, we need some kind of woods for laying and collecting eggs. And as you say in the video, this is the love cage. Inside there is some woods for laying adult insects. And then we collect eggs from this wood and put it in the bag, which mix with substrate. So this is the all product after collecting the eggs from woods. The eggs are put on the substrate and then mixed with the agricultural byproducts. After, of course, these byproducts are grinded in small pieces to facilitate the eating from larvae. And then larvae, they grow until pupa stage, which we put inside the dark cage, and the process is started. Before starting our experiment, we made some assessment with the local market in order to know how quantity of byproducts are generated or produced by markets. So this figure showed that there is many various byproducts, especially from vegetables, fruit, or mixture of both of them, fruit and vegetables, and the variation is between 10 and 300 kilogram per week. We collected then all byproducts from markets. This is an example of fresh byproducts. For example, green pepper, banana, eggplant, carrots, apple, etc. But also we think to collect some dry byproducts in order to make the storage, the long-term storage, very easy. So the main ingredient analyzed are dry matter, crude protein, either extract, ash, and gross energy, in order to know the nutritional value of each ingredient and each byproduct. And then in collaboration with our partner in University of Torino in Italy, they suggest for us this 4 formulation in addition to the control. The ingredients are water, wheat bran, maize, alfalfa, cattle and feed pellets, and potato and bread waste. So the first result showed that the diet formulation number 4 showed the high weight gain and growth rate for larvae. But also there is diet number 5 and 1. Also, we analyzed the protein contents in the larva. The highest one, it was in formulation diet number 3. Chitin content was high in both of diet formulation 4, 5, and 2, and also ether extract, which was higher in diet formulation 1. Also, we assessed the feed conversion ratio, which was very high, very low in diet formulation number 4, and the bioconversion efficiency corrected for residue was also high for the diet formulation number 4. For the fresh weight of frass, it was higher in the diet 1. And finally, survival rate was very high in diet number 3. So in the diet 4, this diet showed a low feed conversion ratio, that indicate that BCF are efficiently turning feed into biomass, and the high bioconversion or efficiency corrected for residue and use a large proportion of organic waste in successfully incorporated into larva biomass. So after this analysis, we should select one formulation that we will use in the intensive larva production that we will use for poultry trial. According to this radar chart, we selected the formulation diet number 4 because it contains a balanced ingredient. So this formulation was used for growth and slaughter performance and animal welfare. For the experiment design, we used 3 treatments. One treatment with control in which we use only local raw material. The second one using 5% of larva. And the third treatment, we use 10% of larva. As you say in the video, the chicken make some competition to eat and to prefer to eat from larva. And then when they complete the larva, they go to for the raw material. For the data collection, we analyzed growth performance, such as weight gain, average daily feed intake, feed conversion ratio, for slaughter performance, drape loss, cold carcass weight, dressing, and also the pH of meat. This is some main results indicating that treatment when we use larva as a feeding for chicken, they show some good performance, such as weight gain, daily feed intake, feed conversion ratio, which is low for the diet number 3, which is in which you used only 5% of live insect. And the most important result is about the high level of cold carcass weight in this 5% of larva. We also analyzed the pH of meat and the drape loss, which was performed and showed good result in this 2 treatments. So the main result, no negative effect on the growth or slaughter performance. However, larva feed birds show increased value in most of the parameters. The larva feed group show good meat quality reflected by low drip loss value. This is other work in progress to assess animal welfare and meat chemical quality. In addition to the protein, the insect farming produce frass, which is a biofertilizer obtained by separating the residue from harvested black soldier fly during the rearing process. This biofertilizer, it contains many nutrients that are very good for crop growing. So we made some field trials in the framework of 2 projects, Prima projects on barley and onion crop in the framework of Advagromet project, and Weisslöpen Fababin group in the framework of Sepromet project. So the field trial are randomized block. We used 6 treatments. T1 and T2 are the control, positive and negative control, and also different rates of the frass in the other treatment. The results showed that the treatment number 3 and 4 are the best rate of application of frass for Fababin, and for Weisslöpen treatment 3 and 5. If the treatment 5, so we used only the frass without chemical fertilizer. So this is some environmental and socioeconomic benefit of insect farming. The price of larva biomass is 2,000 euro per ton. Exivia about 500,000 euro per ton, and also the frass and the compost, which is between 200 and 600 euro per ton. So this is all from my side. Thank you.
Thank you. Thank you very much for the presentation. You really covered all the aspects of the bioconversion of the insect farming. We have time for one question if there is from the audience. Any? Yes, Laszlo?
Thank you very much. Really impressive work. My question is, you showed the efficiency of feed incorporation or conversion, and in the denominator, there was a difference between the fed feed and the residual feed. Do you have any data? What was the ratio of that, let's say in 1 week, or how much was consumed of that given biomass?
We measure each week. We measure the quantity of feed taken by kitchen, and we each week we readjust the quantity of feed that we should give to the birds. So every week we make an update of this feed intake.
Okay, we discuss later. Okay, thank you.
Okay, I have also one short question. So I've been myself in a short-term scientific mission in the University of Turin, and I was following similar experimentation with the growing of the larva, starting from residue from canteen, from the school. And the main challenges that the team there had was to stabilize, the stabilization of the substrate for the regular growth of the larva, and the texture, because the humidity control of the substrate was hard to manage. So how did you face these challenges, if you had as well?
There's not only this challenge, there are many challenges. In the substrate, we in our case in Morocco, we control only the pH, and we control the pieces of the substrate, and we make this substrate very easy for larva feeding. So when the pH is controlled, maybe the experiment can success. But there's also some other challenges like temperature, like humidity, and also the attractant that allow adult insect to mate in the love cage. This is also a big challenge.
Okay, well, thank you, and good luck for the follow-up of this experimentation. Let's move to the next speaker. I'm inviting now Iga Piasecka Lenartovic, sorry for the pronunciation, from the Warsaw University of Life Science in Poland. Iga is an assistant professor there. And today she's giving a talk about ultrasound-assisted extraction of oils from berry seeds, a sustainable approach to agro-food waste valorization.
Thank you very much. You pronounced my name very well. So as it was said, I'm from Poland, from Warsaw University of Life Sciences, and I would like to talk today about our main focus recently in our lab, the ultrasound-assisted extraction of oils from seeds of fruits, mainly from berry fruits. And my presentation will be divided in some parts. I will introduce you to generally what is the amount of berry waste, especially in Poland. And then I will show you our methods of that valorization. So, okay. The fruit production only in Poland equals to almost 4 million tons per year. And over 25% of that number is processed to obtain juices, jams, some concentrates or wines. And pomasses are the main byproduct from this processing, and they are amounting up to 20% of the origin mass of fruits being processed. And the main ingredient of pomass are seeds. So due to fulfill the circular economy policy, and also because of the high nutritional value of those seeds, and some unique qualitative properties of oil obtained from those seeds, we had an idea to valorize those wastes in such a way to obtain oil. So in terms of alternative extraction methods, we know that conventional ones may contribute to natural environment damage due to high energy and solvent consumption. That's why alternative extraction methods seem to be promising. They involve applying new technologies to maximize the yield and to enhance the quality of oil and minimize that negative impact of environment. Our main focus is ultrasound-assisted extraction, but we also use pulse electric field extraction, microwave-assisted extraction, pressurized liquid solvent extraction, or supercritical solvent extraction. And in terms of this ultrasound-assisted extraction, we previously optimized the process for every raw material that I was using. And then we examine the influence on thermal properties and fatty acid share in obtained oils. And in this particular study that I will be showing the results of today, we also examined some chemical properties of our oil. Just to introduce you with the ultrasound, the effect of ultrasound on plant tissue is shown on this figure. So due to acoustic cavitation, son operation, improved mass transfer is observed in our media. That decreases time of extraction and decreases energy and time consumption, which may contribute to environment saving. In our study, we used two different materials. It was black currant and chokeberry seeds, which were obtained from the pomass. These are popular berry fruits in Poland, which are usually used to obtain juices or jams or concentrates. And ultrasound assisted extraction was done in this ultrasound processor shown on the figure, and extracting medium was hexane used in solid liquid ratio of 1:15, and amplitude and time of extraction were based on previous optimization studies. We chose two of the parameters, which was 60% of amplitude in 12 minutes and 90% of amplitude in 6 minutes. We also ran control extraction and in 2 hours shaking in water bath, and we also used cold pressed oils, which were purchased on a local market to compare. The analytical methods that we used was pressure differential scanning calorimetry, which is a tool to evaluate the oxidative stability of oil. It's some accelerated oil oxidation assay, which is run in high pressure and the oxygen presence. in 120 Celsius degrees. We did GC to examine the profile of fatty acids and also carotenoid content and chlorophyll content in spectrophotometer, as well as ABTS, DPPH, and TPC also in spectrophotometric assays. So, as I told you, this PDSC says if the oil is resistant to oxidation, the longer this oxidation time is on the figure, the better the quality of oil and the better shelf life of oil. So we can see that on the left for chokeberry seed oils, ultrasound assisted extraction was quite good, similar to the control one. However, a cold pressed oil is very low, has very low stability. And in terms of black currant oil, the best one was cold pressed. So different phenomenon here. But ultrasound assisted extraction was in the middle between control one, which has had the lowest oxidative stability. In terms of TPC results, total polyphenol content was higher in the chokeberry seed oils. However, the differences were visible only for the 60% of amplitude and 12 minutes time. Next, antioxidant capacity in ABTS assay and DPPH assay have shown similar differences, that the 60% of amplitude was more efficient in terms of chokeberry oil, and 90% amplitude was more efficient in terms of black currant seed oil. When we are talking about chlorophyll and carotenoid content, you can see the these are not the pictures of oil, but of the extracts which were extracted using methanol to obtain this polyphenol rich fraction, but it presents very well the color of this oil. So you can see that black currant seed oils are much more have much more chlorophyll inside than chokeberry seed oils, but in terms of carotenoid content, it is way different, because chokeberry seed oils had higher amounts of carotenoid content. And also in terms of these two parameters, the most effective was 60% of amplitude in 12 minutes time ultrasound assisted extraction. GC results are shown in the table. So generally, ultrasound extraction doesn't really change the fatty acid profile. You can see that in terms of this linoleic fatty acid, there was increase in chokeberry seed oil extracted using ultrasound. In black currant seed oil, we had this increase in gamma-linolenic acid, fatty acid. The oils, both of them, chokeberry seed oil and black currant seed oil, are very abundant in polyunsaturated fatty acids, with the amount exceeding, in most cases, 70%. And linoleic fatty acid is the most abundant in all of studied oils. So as a conclusion, fruit processing generates large volumes of seed byproducts that are often not used or discarded. And our results show that chokeberry and black currant seeds should be viewed not as a waste, but maybe as valuable agricultural resource. And the study highlights that losses occurring after harvest do not have to be avoided only by reduction, but can also be valorized. Ultrasound assisted extraction proved to be an efficient and environmentally friendly method for recovering high quality of oil from berry seed residues. And ultrasound assisted extraction enables loss reduction by extending the value chain beyond primary fruit use and juice production. those oils that we obtain were rich in polyunsaturated fatty acids, particularly linoleic fatty acid, and have shown high oxidative stability and decent antioxidant capacity, as well as content of polyphenolic compounds and chlorophyll and carotenoid. So ultrasound processing preserved sensitive bioactive compounds, demonstrating that low impact technologies can be integrated into some maybe systems. The results of this of the study has been published like a few days ago, so I haven't included that here yet, but I will do this in the PDF presentation. Thank you very much for your attention, and I invite you to read the publications that I have mentioned about optimization of every single raw material, and that's all.
Thank you for a very nice presentation, and congratulations on the recent publication. Do we have some questions in the room? Yes, we have one from Sebnem on the back.
Thank you very much for your valuable study. First of all, my question is the first one. What is the yield coefficient of PFAS? I mean the oils per wasted biomass, or let's say substrate. And have you compared it for the different non-thermal processes?
So to be honest, we haven't studied the exact yield of PFAS in our oils. The yield of oil is, it depends on the raw material, but for chokeberry it is around 12%. And for blackcurrant, it is some number more, but I don't remember the exact one. Yes. So in terms of the results, that it's 70% of those were PFAS, of those fatty acids were PFAS, then we can say it's somewhere around that.
Yes, I think so.
But to increase the yield of oil general in the So in terms of that optimization that I talked about briefly, we compared to the reference method, which was Soxet extraction, and we managed to be at the 100% almost of the Soxet extraction using only ultrasound. Yeah, there are of course some of those methods. We unfortunately do not have them in our lab. Yeah. Thank you.
Thanks. We have time just for one more question.
Thank you for your presentation. I'm curious about, you told about you applied optimization step, but which software did you use? For optimization, we generally use response surface methodology or other software, but in this, I just see two different variables. And where is your, design? So I'm curious about it.
Okay. Yeah, we use the response surface methodology to optimize, and the two variables were time of the extraction and the amplitude. That's why we only use these two variables in this study as well. And the solvent was hexane as well as the reference one. And our responses in this optimization study were yield and the PDST, so the oxidative stability, to obtain oil with the highest oxidative stability and with the highest yield.
Thank you.
Great, thank you very much.
Thank you.
So now we move on to our last presentation before the coffee break. And the speaker is Barbara Henriquez, who's a researcher at the Polytechnic University of Viseu in Portugal. And she's going to present optimizing fig preservation and valorizing pruning biowaste, drying performance and bioactivity of Ficus carica extracts.
So good morning, everyone. My name is Barbara Henriques, and I'm going to be presenting to you a study made in the University of Aveiro, the Polytechnic University of Viseu, and also in Ancona with Marwa and Gianfranco. So I'm going to be talking to you a little bit about Ficus carica and two problems that exist with its increase in production. So Ficus carica belongs to the Moraceae family, and its top producers are Turkey, Egypt, Morocco, Algeria, and Iran. With the increase in fruit production and tree development, we have two key problems. First of all, the post-harvest losses and unharvested figs. This means that there are a lot of figs that are burned, are not used. And our study thought, why not use these figs and get a new technology that enables us a new drying process that extends shelf life, reduces losses, and adds value for local markets. So then the other problem is tree development and increase in pruning. The increase in pruning leads to biowaste, that are the leaves. Normally, these are burned, which increases green gas emissions. But according to literature, leaves are biomasses rich in bioactive compounds, such as organic acids, flavonoids, etc. So we thought, why not turn these byproducts that right now are waste into high-value bio-based products that can be used in the industry? So regarding our first question, how can we have a new drying technology that is better than the traditional sun drying? We tried both techniques, traditional sun drying and hybrid solar drying, and we had a lot of parameters that we studied. First of all, I'd like to talk about the drying time. As we can see, traditional sun-dried figs took more than 5 days to dry, while hybrid solar-dried figs only took 71 hours. Furthermore, as you can see in the water activity for sun-dried figs was more or less 0.68, while the water activity for hybrid solar-dried figs was 0.63. According to a lot of studies for dried fruits, the range for the water activity should be between 0.60 and 0.65 to make sure they don't have pathogens and they are good for marketing. So as we can see, hybrid solar dried figs have a better water activity. Furthermore, we also tested for total phenolic content and antioxidant activity, and comparing to fresh figs, we can see that hybrid solar dried figs had the higher total phenolic content and antioxidant activity. We also did mass spectrometry for the phenolic compounds, and here we have two results that can be talked about. First of all, fresh figs have a higher content of rutin, which is also the same for hybrid solar dried figs. But regarding the caffeolic quinic acid, we can see that the same trend, high in hybrid solar dried figs, but for the second most present phenolic quercetin derivative, sun-dried figs had a higher content than hybrid solar dried. now we also did another analysis about the microbiologic analysis, because what we need to make sure is that these dried figs are good for marketing. So we need to make sure that they don't develop any pathogens. So we tried several figs, both hybrid solar dried and traditional sun-dried. In figure A, we have fresh figs, and as we can see, after 4 weeks of incubation, they developed several fungi. And the same happened to traditional sun-dried figs, as we can see in B. Figure C is actually hybrid solar dried figs, and after 4 weeks, they had not developed any fungi. For the fresh figs, the fungi that were most present was Cladosporium spp, while for sun-dried figs, it was Alternaria alternata. Then for our second problem, the waste of the leaves. We thought about doing some extracts. So we did several tests and chose 2 types of extracts, one with 70% ethanol and another with 96% ethanol. We extracted them, filtered, and then we froze at minus 20 degrees. We did tests for the total phenolic content and antioxidant activity. We had contradicting results because, as you can see, the extract with 70% ethanol had a higher phenol content, which would probably mean that it had a higher antioxidant activity, which was not what is shown. It's shown that the leaf with 96% ethanol actually had a higher antioxidant activity. So we decided to try both of them. And where did we try them? We tried them in a species called Tuta absoluta. So this was, as I previously mentioned, was a study made with the agrarian school in Viseu, and they have a big problem with Tuta absoluta in their crops. This is a species that was originated in South America, but has quickly spread to Europe, Africa, and Asia. And it's mainly a threat to tomato crops, but can also occur in potatoes, wild tomatoes, eggplants, and apples. Unfortunately, synthetic insecticides, have been having some problems, like the resistance of the species to the insecticides and also control of failures. So producing botanical insecticides with the extract of the leaf was our theory to lower the environmental toxicity, and also because then we can produce compounds that have defense mechanisms against pathogens, herbivores, and predators. To do this experiment, we took some leaves from the crops in Viseu. We dipped them in each of the extracts, put them in petri dishes, and added some larvae to the petri dishes, recording the mortality after 24, 48, and 72 hours. As we can see in this table, the only one that had a statistical difference was the Ficus carica extract with 70% ethanol. It had a statistical difference against the control, and this shows that the application of this extract has a higher caterpillar mortality when compared to the control, both for the 24, 48, and 72 hours. Then we did another test against Botrytis cinerea. I took the 70% extract to Italy, and we did in vitro and in vivo testing. For the in vitro testing, we did 2 solutions, one with PDA plus treatment and another with just PDA. We put them in the petri dishes with Botrytis, and then we saw what happened every day during 5 days. For the in vivo testing, we used grapes. We did a small hole in each of them, and we tested 2 concentrations, a 1% concentration with the extract and a 10% concentration. And once again, we controlled the growth of Botrytis. Unfortunately, as you can see in these graphics, there was not a statistical difference in either in vivo or in vitro testing. The conclusions that we can take from this study is that the hybrid solar drying system proved to be an effective alternative to traditional sun drying, especially in the drying time and in the water activity that is very important for marketers. Also, dried figs retain relevant phenolic compounds, and this make them very good for selling. Regarding the application of the fig leaf extract, the 70% ethanolic had a statistical difference and the 96% did not. And also the 70% ethanolic extract did not have an in vitro and in vivo statistical difference, but also we could be testing different concentrations because we were short on the amount. I only took a little amount to Italy, so we couldn't try higher concentration extracts. These results highlight the relevance of optimization of this extraction strategies and compound purification, enhancing bioinsecticide efficacy, reinforcing the potential of fig byproducts, and with sustainable framework valorization. Thank you for your attention, and I'd like to thank my supervisors who could not be here, and Gianfranco and Marwa.
Thank you very much, Barbara, for your presentation and for keeping the time. So we have time for questions if anyone want to ask. Yes.
In each of the Petri plates, we did have more than one species of fungi, and we identified each of them. And also we took a little bit of each of the fungi and put them in smaller Petri dishes for it to grow and make sure of the mycotoxins present.
Because you know that in dry, this type of fruit, actually in dry figs, there is commonly release of mycotoxin. Maybe
in the new method, we tested for 4 weeks. These are the results for 4 weeks, but we also left them for the time I was in Italy, which was 8 weeks, and we did not see the growth of any fungi, and we tested, I think, 20 figs. I think it was around 20 figs with the new method, and we did not see growth in any of them. So for now, I'm believing that this is a safer way, but I do think there is a need for further testing.
Okay, thank you. Any more questions? Yeah.
I'm just curious, is it just frequency you look at, or did you look at covariance? You know, any of the statistician measures?
We only check for the frequency, yeah.
So just how frequently?
Yeah, because
so it's statistical significance that you're measuring.
Yeah, because I saw some statistical difference for the extracts of 70%, but unfortunately, we did not believe that it was enough of a difference to keep repeating the test, especially because this was made in collaboration with a colleague who's taking his master's degree, and he had to deliver his thesis.
Okay, good.
Okay, thank you. If there are no more questions, we have one more. Yeah, the last one, yes.
Sorry, I couldn't
understand me. I mean, white latex of the
we did not. No, we did not include the. No, we did not include the latex because. Yeah, I know, but there are already a lot of studies about the latex. Our study was trying to be different, but yeah, we studied a lot before deciding what to test.
Okay, thank you again then. And so we are moving to the coffee break now. We have half an hour of break. There is all the poster session still there, so you can use this time also to go and check on it. I kindly ask the speaker of the session after the coffee break to upload the presentation if you haven't done yet, and then see you here back at 11.
Name is Stanchuk. It's a difficult name to pronounce. First, I would like to thank the organizer for giving me this opportunity to speak about a national project with a very long name. I'm a professor at the Faculty of Food Science and Engineering, Dunărea de Jos University of Galați, which is situated in the southeast of Romania. It took like 3 hours to reach the capital. So I'm the manager of this project with the acronym SAFIR, Sustainable Alternative Food Resources for Future Food by Widening Innovation into New Composites with Improved Health Promoting Properties. It doesn't work. What should I? Okay, so what should I do? Just the, okay. This project is founded by the Executive Unit for Financing Higher Education, Research, Development and Innovation, and implemented by our university for a 3-year period. I would like to talk a little bit about the current status in Romania very shortly, because in Romania is like in Turkey. You heard the presentation from yesterday. We have a national strategy for sustainable development and a lot of regulation regarding the management of waste. For example, for grape pomace, I found a regulation which oblige the producers to use the pomace as feed, composting oil, exports, incineration, delivery to an authorized landfill and others. However, the term byproduct or co-product, as I heard yesterday, was recently implemented in Romania in one of government regulation, and it is, for me personally, it is questionable if Romania will be able to reach this percent of degrees with 50% by 2030 regarding the management of waste. Also, there is a huge gap between academia and economical practices in Romania, so we need more innovation in order to increase the sustainable use of waste and byproducts, and also to sustain the technological transfer. So this project starts with several aspects regarding, based on FAO estimation, 1.5 billion tons of fruits and vegetables are produced annually, from which 0.5 are either wasted or treated as by products. So we have sustainability issues regarding efficient food system for the sustainable use of these resources. And SAFIR wants to facilitate the transition from waste to wealth by valorization of selected byproducts into multifunctional ingredients through flexible and interconnected bioprocesses. And at the end of the project, we hope to deliver some tailored recovery procedures for several bioactives like natural antioxidants, antimicrobial agents, vitamins, proteins, starch, pigments, and so on. So the project is focused on food byproducts, and we selected 4: grape pomace, apple pomace, whey, and brewery spent grain for developing cross-cutting actions which are common across various stages of food value chain and are needed to achieve global food loss and waste targets. And the project uses methodology, extraction, encapsulation, drying, and fermentation to provide in the end some multifunctional powders to be used in different hybrid foods. At scientific level, we managed until now to identify some natural competitive inhibitors for different enzymes. This year, we are going to use to develop new alternatives for whey valorization as a source of lactose and proteins for co-crystallization and encapsulation, micro and nano encapsulation of bioactives from grape and apple pomace, to fostering the micro encapsulation methods to deliver delivery targeted bioactive, to reuse and recycle the whey and brewery spent grain as easy to use strategies from transforming waste into postbiotics. Using probiotics as culture, to develop novel multifunctional ingredients with added value and functional potential, and as I told you, to develop technologies for hybrid bakery and meat products with the aim of replacing meat, fat, or flour with selected powders. I focused my presentation on grape pomace because we had a lot of results from the first year of founding, and this is the current situation in Romania regarding winery. We have 611 companies with objective of activity, wine production and grape cultivation, and we produce like 1 million of tons grape per year, so a huge amount of grape pomace is obtained. In the first year, as I told you, we tested new alternatives for phytochemical extraction from grape pomace and apple pomace. We used NADES as eco-friendly solvents. NADES are a mixture of 2 or more compounds in a certain molar ratio, basically a hydrogen donor and hydrogen acceptor. When heating this solution, a decrease in melting point took place, and you have liquid solvent ready to be used in extraction. In each experiment, we used response surface methods and central composite design to optimize and validate the ultrasound assisted extraction of bioactives from red, white, and apple pomace. And we obtained enriched extracts, especially in phenolic acid. For example, red grape pomace was used as available source of biomolecules with inhibitory effect on pro-inflammatory and metabolic syndrome associated enzymes. This validated extract we identified 32 polyphenolic compounds with a total polyphenolic compounds more than 500 milligram per kilogram. Mainly, as I told you, phenolic acids and a high quantity of vanillic acid in the extract. So we tested in vitro the inhibitory effect on lipoxygenase, alpha amylase, and alpha glucosidase, and we obtain a high rate of inhibition. In silico investigation show us that for alpha amylase, vanillic acids is the main phenolic acid which has the affinity to bind to the active site of enzymes. Suberic acid did the same thing for alpha glucosidase, and in case of lipoxygenase, suberic acid and the resveratrol were the main compounds binding to the active site of binding site of enzyme. These bioinformatic methods allow us to identify the amino acids involved in interaction, but the types of nature of the interaction between the ligand and the protein. Also, we use the same statistical analysis to optimize the extraction of polyphenols from white grape pomace, and we tested this extract as an inhibitor for polyphenol oxidase. The extract based on choline chloride and lactic acid in a molar ratio of 1:2 allowed to obtain an extract with 23 compounds, and of which approximately 70% were phenolic acids. By experimental methods, we obtain an inhibitory activity of more than 40%, and the in silico studies show us that polyphenol oxidase has 2 binding sites for the compounds, 1 binding site for vanillic or suberic acids, and 1 binding site for polydatin, fluorescein, azelaic acid, chlorogenic acid, and resveratrol. So these extracts can be used to prevent the enzymatic browning in some products. In case of apple pomace, we dried. We used 2 drying methods: hot air convection and infrared drying method based on drying kinetics, color parameters, and polyphenolic content. And also we optimize the extraction using choline chloride and glycerol in this case in a 1:1 molar ratio. We obtained an extract with catechin and epicalocatechin in high concentration, followed by phenolic acids, mainly protocatechoic acid. And you can see here some pictures from dried apple pomace. We correlate the drying kinetics, especially with color parameters.
Sorry, Nicoletta, you just have 30 seconds to finalize.
I am going to finish.
Perfect, yes.
So we combine red pomace and apple pomace to dry, and we obtained based on the same statistical method, and we obtain a best ratio between grape and apple pomace of 1:3, dried at 70 degrees by convective method. And we also calculated the bioaccessibility index, and you can see here a high bioaccessibility index for anthocyanins. Next, we are going to use this extract in nano and micro encapsulation using proteins from whey in combination with different biopolymers. And we want also to formulate a culture media for brewery spent grains to obtain postbiotics, and in the final to obtain powders and hybrid food. So thank you for your attention, and I'm sorry that I talk too much. You can, I should tell you that in 1 year of funding, we have 4 articles, one article is published, 4 under revision 1, and 1 it will be ready to submit at the end of February. Yes, it's a very good project and fulfills completely with the aim of this project.
Thank you very much, Nicoletta. And yeah, it is a great project. Unfortunately, we have no time for questions. So if you have questions, you'll have to catch Nicoletta in the lunch break. Okay, so thank you. So we're moving on to our final presentation by Jovana Pantic, who's a research assistant at the University of Novi Sad in Serbia. And her presentation is on from waste to functional materials, biopolymer material synthesis in the framework of a circular economy.
Hello, everyone. My name is Jovana Pantic. I come from Novi Sad, from Faculty of Technology. Today, in this presentation, we will start from a different kind of waste, from plastic and from packaging waste. And after that, later on, we will cross the discussion with agro waste as well. 1/3 of all produced plastic is dedicated to packaging materials, mostly one-time used items that are immediately discarded after consumption. So those wastes is generating massive amount of waste and cause harmful effects in ecosystems. So it is mandatory to minimize the appearance of plastic oceans, rivers, landfills, and to protect suffering wildlife from eating plastic. So what are solutions? One of solutions are biopolymers. I like to call them materials of green era, especially in food packaging, because they are biodegradable, sustainable, and eco-friendly. Their production relies on renewable sources. Non-toxicity is also crucial, as it ensures safety for both human health and environment, and they are cost-effective, since they reduce costs related to recycling and waste management. When it comes to agro waste and on the other hand, its valorization, it is good to say that there are many modern technologies that tend to valorize various agro waste in terms of converting to other products. This is the situation where we convert the agro waste into biomaterials and thus achieving double green effect. So what falls inside this double green effect? First of all, there is a resource optimization and creation of new value. Then instead of landfilling, agro-industrial waste is converted to other products like bioenergy, packaging, composting, animal feed, biochemicals. Further, it contributes to environmental protection and economic sustainability through innovative technologies and circular businesses models, and it fosters innovation by applying new technologies to waste processing. When we talk about valorization, it's important, it makes sense when we have a huge amount of agro-waste. So it's important to say that agricultural and food industry generate massive amount of waste with estimates from 2 to 3 billion of tons per year. And these waste are produced at every stage of within the food supply chain. But inside of primary production and processing manufacturing, together, those stages together make up about 49%. The leaders inside this group are the oil crops and fruits, and after oil extraction, there are various byproducts, but the most important for us are oil cakes. So those byproducts are extremely valuable, and they can be converted into other value-added products using many new technologies. And in addition, the reason why we should improve the valorization strategies is the rising amount of these byproducts over the last 20 plus years. Among various traditional oil seeds that we were recently focused on, we now shifted our attention to plum seed, which is not typical oil seed as well. The first reason is plum availability in Serbia, because there are a lot of plums in Serbia, because it ranks third. Serbia ranks third in the world in plum production, right after China and Romania, and it is most widely grown crop in Serbia. And the second reason, so when there are a lot of plums, there are a lot of byproducts. So before discussing nutritional value, we should consider some numbers again. But although there are not statistical data, those are some theoretically estimations from the literature. So plum pits account for about 16% of the fruit mass, that's generating about 78,000 tons of plum pits in Serbia. And given that the seed accounts for 9 to 30% of the plum pit mass, this corresponds to approximately 7 to 23,000 tons of plum seeds per year in Serbia. So if we waste all of those amounts of seed, then we will waste macronutrients like proteins, carbohydrates, oils, dietary fibers, and micronutrients as well, like total phenolics and total flavonoids. So instead of waste them, we should find the valorization strategies. And one of the possible ways are protein-related products. And on the other hand, a great amount of oil in plum oil cake suggests oil extraction. And plum oil is nutritiously rich, and it has many health-related benefits. But after oil separation, we have the new remaining byproduct. Actually, the oil cake is the byproduct of the byproduct. So instead to waste them, we could valorize them into the food packaging. How we can do that? There are two main approaches to make biofilms and biocoatings. In the case of biofilms, they are typically produced in the lab by casting method, where we dissolve or disperse biopolymer in the water, then add plasticizer, and then we adjust processing condition like pH and temperature. And after that, film forming solution is filtered, poured onto the flat surface, and dried. And after synthesized, biofilms are ready for application. Application can go into two directions. The first one is to form bio bags, like here in the picture. We package the sugar inside, so we can make stand-alone bio bags if the materials is heat sealable. If it's not, then we can help it by adding the new biopolymer material that is heat sealable. And here in this video is presented the lab scale synthesis and its characterization in short. Great amount of agro-waste is underutilized, such as oil cakes. Of particular importance are oilseed cakes obtained by cold pressing without the use of organic solvent. So, oil cake valorization through biomaterials production makes double green effect, following green chemistry principles: renewable source, low cost and safe process, easy to handle, free of pollutants, not requiring high pressures and temperatures. So, it was the presentation of lab scale process. And here is a bit different approach where we coated the commercial plastics polyethylene with palm oil cake based film. And we managed to heat seal the polypropylene containers filled with protein plum jelly. So we are so proud of this product. We managed to do this in industrial conditions, and we can say the product is almost all plummy. And besides plum oil cake, there are other oil cakes that can be converted into functional materials like pumpkin seed oil cake, sunflower, rapeseed, and flaxseed. They all can form functional biomaterials ready for application, but where they already demonstrated its effectiveness is this example where flaxseed oil is packed inside the pumpkin oil cake based bags, and we managed to successfully to prolong the shelf life of the packed oil. In this approach, sunflower oil cake was extruded to these biomaterials. And another approach for applying biopolymers is edible coatings, as some of the colleagues mentioned yesterday. So there are typical coating deposition methods like dipping, cross-linking, and the spray coating. We use in our recent studies where we coat grape with flaxseed oil cake based coating, and the fresh cut apples we coated with plum oil cake based coating, and we managed to prolong the fruit shelf life that is extremely susceptible to deterioration. And maybe you're asking yourself why those oil cakes are so attractive. They are highly suitable for zero waste, to try to reach zero waste concept, because they can be converted with minimal to no waste streams. And those are some publications that provide evidence to this approach. And here is the case study of the closing the loop, where the starting point is agro waste, actually plum pit, that then from where we recovered the seed, and using various green methods, let's say, we recovered proteins, green energy, biopolymers, or pharmaceuticals, or even if we decide to obtain the oil, after that we have new entry point, that is plum seed oil cake, from where we make biofilm, then we pack the product inside. When we use that product, there is a biopolymer waste, we can recycle it, or if we are irresponsible, and if we waste it into surrounding, they are compostable, and it won't pose any burden to the environment, or we can convert it into biofuels. And so that was the one effective example how we can close the loop with the plum. But what is the most important, that this concept is not limited just to plum. It can be successfully extended to many other agro-waste feedstock, as long as we understand the system properly. So what really matters and where are the key challenges comes down to 3 points. The first one is agro-waste feedstocks. We should understand its chemical composition and to know what to expect of this agro-waste. We would like to have a low price of it. And the second, the most important thing is biomaterial properties. So we should obtain functional biomaterials with satisfying mechanical and barrier properties that will be compatible for application. Application can be food related or non-food related applications like If we choose food, there are a lot of fruits, vegetables, meat, cheese, and sweet or non-food related application, cosmetics, pharmaceuticals, and others. So there is a lot of space for creative science, with the aim of achieving this zero waste concept. If we efficiently mix chemistry, materials functionality, and application. Thank you for your attention. If you have any questions.
Thank you very much, Ioana. Very great job you have done.
Thank you.
We have time for only one question, actually. So if there is anyone from the audience that has a question. Seems no, then, oh yes, we have one.
Thank you for your presentation. Very nice and very interesting. I was very curious, why do you call your materials biofilms? Because biofilms means something else. They are produced by microorganisms and are very hardly to remove from equipment in the food industry. I'm a little bit confused. Maybe you can explain better.
Yes, actually you are right. Those are biopolymer films or bio-based films. And in some literature, there is no strict limitation to call them biopolymer films. So regulation is the one that is missing to better understand those terms. So from this point of view, I can call them biofilms, but I understand what you think about.
Okay, thank you very much. And I close the session of the working group 4. I'm happy about the presentation that we were able to screen. Thank you for all the speakers that were attending and presenting here. Thank you for all the questions. And now I'm leaving the place to the working group 6. And Kata, the leader of the working group, who is coming to present the activity done within the group.
We are at last, but not least, of working groups, because whatever we do, we have to communicate. And you are doing in an excellent way. I saw that some of you posted some pictures of the meeting, and some of them got hundreds of likes. So congratulations, you are the best communicators. Together with this, that is important, and everything is also the pictures that were taken yesterday by the official person of the university are now on Facebook page, so you can see yourself. And also all the video are there and is in progress. People are sharing, and that's important. You can see yourself and see your colleague and share with your colleague. But now let's see what years ago we placed into the program, and now we have to do for UG6. Kata, UG6 leader, the floor is yours.
Thank you. So hi everyone again. Yeah, so here you can see the members of UG6. We have a really nice numbers, which is good because what Gianfranco mentioned, it's really good that we have really nice results, but if we don't share it, it will be useless. But what about working group 6? Just highlight that it's about the networking, the dissemination, the communication, and we will disseminate the results and the findings of the other working groups, what they already did and will did will do. And we will have the communication with the main stakeholders, and we need to deliver the provided tools and the recommendations. And of course, we have several activities, what you can see here. And we have only 2 deliverables, but this 2 is really important. So what we do until now, when we start this project, first we need a website. So this was the first what we did. And of course, we need different social media platforms. And from the first years, we organized several STSM, and it's really nice to see that how many young researchers from ETC can join different research work. So if you have possibility, you can apply for this STSM. And we organized already 2 training schools, and next will be in Latvia. And the last year there was 2 webinars in WUGI 4 and WUGI 6. And of course, the conference is really important to meet and do networking. And here you can see the 1 deliverables. So we have the website, is good, but we need to do this website with different languages. And in the table, you can see how many translations we have, already 20. And here I would like to thank you, the members who helped us to do this translation. But you can see that we have some missing language. So if you have a little free time and you have a capacity, motivation, please help our work to translate a short text about the cost action and about the working groups. The translation that we have is uploaded, are in progress. So I think within 1 week you will find in the websites. And the other activities and deliverables that need to do, but we have time because we have to finish it in the last year, that the findings of the other working groups, we will do different protocols, papers, the best practices, and guidelines or training with those, and we have to upload it, upload to the website, and of course share in different way. And after in the WUGI 6 meetings, we will talk about it, how we can do this. So thank you for your attention. That's all.
Thank you, Kata, for this presentation. I don't know if you have any question to Kata that are for the general audience, because later we will go in the WUGI meetings. Do you have any question for Kata or any proposal on how communication can be improved? Okay, we will have time to discuss about, as occurred for the other working groups, so WUGI number 1, 2, 3, and 4, also for WUGI 5 and 6, that are the one that will provide the fruits later in the time frame of the action, it will be circulated soon a link in which you can contribute to the deliverables, as was done for the previous deliverables. And then you can provide your inputs and you can join the different activities. So it will be the chance to apply. If you want, you can do, you can contribute, as you are doing in an excellent way for all the other WUGI. Thanks to all the people that collected the translation of the website in how many languages? 20 languages. So some they will be online likely next week, but we circulated even for that the request for cooperation, and a good bunch of people that are now here in the audience contributed. So thanks to you. In case your language is not in the list, your national language is not in the list, you can mail Kata and we can make a further update of this list. So we are going very well with the program, and even the end of the meeting, we can try to make a little bit shorter because a list of things are going on. Even from the lunch, we can come back a little bit earlier. It can be wise. So to start with the General Assembly, if we can recover 15 minutes, so instead of 2:30, it can be 2:15. It can be a good timing. Or if there are not so long line, even 2 o'clock. So try to be back as early as possible, because earlier we start and earlier we finish altogether, because I know that several of you will be back and start to travel. So earlier we start and better it is. But it will be great to resume what it is cooking in the different work groups. So we will have some kind of updates by the WUJI leader about the discussion that will occur, and a kind of timetable of what it is going from now to the end of the project. So into the program, we had a presentation, but we agreed to not have the presentation online. So we thank Paraschevi for the availability to join the meeting at the beginning, but later it was all the people that were invited, to those people was given the opportunity to present, but just to the people that were invited and are here in Zadar. So are not planned online presentation. So just the people that are here are allowed, and just the people that are here are covered by reimbursement. It was not a joke what I told yesterday, that you have to place your signature close to your name. Because even today, we realized that several of you didn't place the signature close to the whole name. If you have been invited, there is your name printed, so you don't have to place on the last page. Cost does not take in consideration that one. So be aware that if you don't get the reimbursement, it means that you didn't sign in the proper place. It looks a very simple issue, but maybe not simple for you from tomorrow on. Okay, so try to be aware about this, and in case, double check. There are a few cases that still to be fixed, and I wish you that are fixed within the end of the meeting. Okay, so now we are saving some time, and Slavan, that did a very good planning, very good job, will announce how we will split for the parallel meetings.
Okay, the VG4, we saw also from the presentation, is the most represented group, so they're staying here. VG1 will follow George. I showed George where the room is. Now we have to open it. And I hope that my students are back. I haven't seen them a few seconds ago, but okay, can be something. Then let's say VG3 is going to the room, that one there, but the entrance is where the table was, one of the tables. You will see it's 121, I think. The other group is going, so here is VG3, VG4, VG4 is here, VG5 and VG6 is going to this room. So just turn the corner, it's 124. And VG2 will follow me. We are going in the basement. And I also would kindly ask all of you by the end of the lunch or by the end of the meeting, start to remove your posters. So we were kindly asked to do that because this is the rector's space for his events, etc., and he's having something this evening, so they have also to prepare. So let's help them. Thank you.
Thank you, Slaven. So the different working group will have this kind of parallel meetings. It is supposed that they will close at 1 o'clock, even it is 5 minutes earlier or later. The different people can go directly to the cafeteria, to the restaurant where we were yesterday. So let's meet there, and then let's meet back here around 2 o'clock. So the online session, I thank all the people that today and yesterday followed online. The online session closes now, and we will open again around 2 o'clock. So thanks you all, and see you later.